Greeting friends, in the last video we talked about what is chirality or asymmetry in carbohydrates, especially monosaccharides. In this video we would talk about what is the effect of this chirality. We know if there is chirality then there would be stereoisomers, and in this video we would talk about one of those stereoisomers, that is the enantiomers. So what are enantiomers? These are the mirror images of each other, the isomeric forms of these enantiomers, the different forms of a compound, that is the enantiomers, are mirror images of each other which are non-superimposable. What does this mean? It simply means you can take both of your hands, and this is your left hand, this is your right hand. Okay, these are mirror images of each other, and your left hand is non-superimposable on your right hand this way. That is human hands macroscopic analog of this property. Okay, these have same physical properties except for the direction in which they rotate plane polarized light. But despite of having same physical properties, they have different biological properties. So now this is the point we are interested in, that is the different biological effects. That means there is a difference in the biological properties of two enantiomers of a compound. This is the property which we are going to exploit, and we are going to talk about this property in the future video. Okay, now there is a key point, and that is all the monosaccharides except a particular monosaccharide which is a keto sugar, that is dihydroxyacetone. This is the molecule, this is a compound which does not contain any chiral carbon. Why it does not contain any chiral carbon? You can see here in this structure, because the atoms or the group of atoms attached to it are not different, and in this case the four atoms or the four group of atoms are different. CH2OH, CHO, OH, H, all are different. Here these are not different, and we know there is a condition for a molecule to be chiral, that all the four atoms or group of atoms attached to it must be different. So this is the one molecule in all monosaccharides which is not chiral. Remember this point. Now there is a system to name the monosaccharides, and that system is D and L system of monosaccharide. To name these monosaccharides there is a system which is D and L. These D and L letters denote something, and that refers to the configurational stereochemistry of molecule. Configurational stereochemistry means the 3D arrangement of atoms. Okay, now what does this mean? We are going to discuss. Okay, so before that we must know there is a key convention, and what is that? One of the two enantiomers of glyceraldehyde must be designated as D-glyceraldehyde and the other one as L-glyceraldehyde. You can see here, this one is the D-glyceraldehyde, where here, okay, and this one is the L-glyceraldehyde. There is a difference in these two molecules. The hydroxyl group here is on the right side, and the hydroxyl group here is on the left side. How we are going to use it? This is the question. This is just a reference compound. Okay, and using this reference compound we are going to name other monosaccharides which are higher in containing the number of the carbon atoms. Here these are 3 and they would be containing more. Okay, now how to know if the particular given compound is D or L? So for that we can group them into two. Group these enantiomers into two, that is, see here, these enantiomers can be divided into two groups that differ in the configuration about the chiral carbon. configuration about the chiral carbon, listen to this point, that is most distant from the carbonyl carbon. You understand? See here, the chiral carbon here most distant from the carbonyl carbon. This we know is the carbonyl carbon, and this one is the chiral carbon. So this chiral carbon which is most distant or farthest from the carbonyl carbon. Okay, now we can elaborate it more. See here, those in which the configuration at this reference carbon, this one, okay, is the same as that of diglyceraldehyde, are referred to as disaccharides, in this case D-glucose. And if those with the same that of L-glyceraldehyde, that would be referred as what? Allosaccharides, or in this case L-glucose. We make it more simple. In other words, when the hydroxyl group, okay, when the hydroxyl group on the reference carbon is on the right side, then it would be referred to as disaccharide or D-glucose in this case, compared to this molecule. Okay, and if it is on the left side here, then it would be referred to as L-saccharide, in this case L-glucose. See the reference compound, L-glyceraldehyde. Okay, now there is a very important distinction point to note. Don't get confused. Confused with what? There is one more terminology here, that is small d and small l. What do we mean by this? It means dextrorotatory and levorotatory. This capital D and capital L are entirely different from this small d and small l, because this is something related to optical rotation. Small d means the compound which rotate the plane polarized light towards right, and small l means the compound which would rotate the plane polarized light towards left. How this is different, this capital D and capital L? Because it is determined on the basis of anomeric carbon first, and how its orientation compared to glyceraldehyde. That is, if this is on the right side, it would be D-monosaccharide, and if it is on the left side, simple, L-monosaccharide, in this case D-glucose or L-glucose. I think it will help. Thank you.